As Michelle mentioned, my name is Tamir, and I'm a neuroscientist. I'm not a clinician in this group. I'm part of the imaging group led by Dr. Klein, by Johannes. And my previous research work, before joining the team, I focused on investigating brain and cognitive resilience in both healthy and pathological brain processes during aging, which is very relevant and, of course, naturally linked to what we are doing in this entire project, and also as a result of physical exercise. Okay, so brain MRI in the OPDC and in general enables us to get a non-invasive view, most of the time non-invasive view, and insights into the living human brain. And from the MRI scanner, in this case, we can get different pretty pictures and images of the brain. Each one highlights different aspects and properties of the underlying tissue, in our case, the brain. And by using different analysis methods, we are able to extract different meaningful pieces of information regarding the brain. For example, regarding different regions in the brain that we know or suspect that are more vulnerable to the pathology of the disease. And for example, take those brain regions and map their physical and functional connections to different other regions of the brain, and to see how those are compromised during the course of the disease. And by using those imaging data, we basically aim to accomplish 4 main goals. The first is to understand the mechanism of the disease. How is the brain affected and changed over the course of the disease? What is the biology that underlies the changes that it undergoes in the brain during the disease? The second is to be able to predict future disease states. For example, who is more likely to develop the disease? Who is more likely to develop complications? What will be the rate of progression of an individual? What is the prognosis? The third major aim is to be able to identify and stratify and select patients or individuals who, for example, will be most likely to benefit from a new clinical trial, from a new treatment, from a new therapy or drug. And also to use our imaging markers or imaging measures as endpoints or outcomes in those clinical trials. For example, to see if a new treatment or therapy has worked or not. And of course, this is in addition to the obvious clinical measurements, as mentioned before, are more noisy and more susceptible to day by day and even hour by hour fluctuations, for example, in our mood or motivation, how I slept in the night, or whether my wife yelled at me before I left the house in the morning. Okay, so the most well-known brain system that is affected in Parkinson's disease is the dopamine system, which originates in an area in the brainstem called the substantia nigra. You can see it in the left here, the substantia nigra of a healthy individual, and here in Parkinson's disease, when we lose those pigmented dopamine cells. And we can also see it on the right using a sensitive MRI modality. Thanks to your participation, this is based on your MRI scans. And we know that dopamine is mainly associated with the motor symptoms of Parkinson's disease. However, we know that patients with Parkinson's present a variety of symptoms, both motor and non-motor. And dopamine dysfunction itself cannot account for all of these different symptoms. Therefore, a main goal of our research, and especially the imaging team, is to try to understand how other brain systems, how other chemical brain systems are also affected during the course of the disease. And one of those systems is the acetylcholine system. Acetylcholine is another neurotransmitter, another chemical, a very important chemical in the brain. It's also called the cholinergic system. And it is located in the deep middle of the brain in an area called the basal forebrain, which is about here in this scheme of the anatomy of the brain. And this molecule, acetylcholine, has many vital brain subserve many vital brain functions. For example, it helps to keep brain cells healthy and functioning. It also mediates cognitive functions such as attention and learning and memory, and some physiological functions such as regulating sleep cycles. And all of those functions are compromised in some way during the course of Parkinson's. And an interesting fact, an interesting finding regarding this cholinergic system is that it has been shown to be affected pretty much simultaneously at the same time as the dopamine system in the brain. Could start even years before the diagnosis of actual Parkinson's disease. And while we mentioned just briefly that dopamine is mainly associated with the motor features of Parkinson's, the cholinergic system has been associated with other different known motor symptoms of Parkinson's, for example, such as cognitive function, which relates to the previous talk, also to aspects of mental health, such as mood, and also to psychiatric features. And now, just to give a few examples of how we address the main aims, the four main aims I've just talked about a few minutes ago, here's an example from a mechanistic aspect or mechanistic point of view. Currently, the gold standard way or approach to examine or assess the function of the cholinergic system is to use PET scans. However, PET scans are both very expensive, also include injection of radioactive substances, and in the UK, from what I understand, it's very, very hard to get approval to use PET scans in clinical research. And using PET scan in previous works, it has been shown that in Parkinson's disease, the main dysfunction occurs in the back side of the brain. We can see here in blue. This is the back, and up is the anterior, the front of the brain. However, we almost can't use it in research. So thanks to your participation and your scans and your imaging data, we've been able to replicate, to identify the same patterns as in PET scan, but here in functional MRI. We can see a very similar spatial pattern of dysfunction of the cholinergic system in Parkinson's disease. This time, non-invasively, without any harmful radiation. Another example from a predictive perspective, here using another modality of MRI, another technique called diffusion MRI, we've been able to reconstruct the physical connections between this basal forebrain region in which the bodies of these cholinergic cells lie, and its physical connection with the other parts of the brain, which you can see in blue. And what we found is that greater structural integrity of those physical connections was associated with better cognitive performance years later in already diagnosed patients. However, in addition, we also found that this marker has a predictive potential even years before Parkinson's even diagnosed. We found that individuals with REM sleep behavior disorder, with RBD, who eventually developed Parkinson's disease, greater or increased structural integrity of those physical connections of the cholinergic system was associated with longer time until Parkinson's diagnosis. They were able to postpone their, let's say, disease progression or disease onset to a more late stage. And just to wrap up those examples with all of our main goals with our cholinergic system, when we're talking about mechanism, we've identified a proposed vulnerable brain system with all its subcomponents. Now we would like to use those imaging markers that we identified as vulnerable in this disease in order to be able to predict future disease states. For example, who will be diagnosed earlier or later, what will be the future prognosis. For example, future complications, or on the other side, maybe clinical resilience to the disease. Next, we use those markers to identify and select patients based on those markers, for example, for a new clinical trial that aims to test a cholinergic drug, for example, a drug to try to address this specific brain system that we know that its function is compromised during the course of the disease. And in the end, after we've selected patients, we use imaging markers of this system in order to assess whether, for example, the therapy of the new drug was successful or not. And I'll just leave you with this little video of the reconstructing system that we have found that overlaid on the entire connection system of the brain, the white matter of the brain. Thank you very much for listening.